My name is Feng. I'm a research leader at Griffith Institute for Drug Discovery. My research is really focused on chemical biology of natural products and more in recent years about traditional Chinese medicine. We're looking at really using molecules from nature to use chemical tools to study the disease biology for Parkinson's disease.
Parkinson's disease is a degenerative disease of the brain, and it affects about 80,000 Australians. And it's estimated to cost our economy around $10 billion annually. I'm George Mellick, the lead in the Queensland Parkinsons Project. There are currently no treatments to prevent, to reverse, or to cure the brain death that we see in Parkinson's disease. As our population increases, it's expected that there will be double the prevalence of Parkinson's within the next 20 years.
I'm Stephen Wood. I'm a Principal Research Fellow. Our group works on Parkinson's. We have people who are epidemiologists that look at population movements of Parkinson's and incidence of Parkinson's. We work with clinicians. We also have geneticists, so we do a lot of the Queensland Parkinson's projects involved in looking at familial or inherited forms of Parkinson's disease, and that way we discover new genes that might give clues into novel aspects of Parkinson's disease. And then that work filters through to my expertise, which is we take cells from these patients and we look at the cellular or molecular mechanism underlying the disease in that individual.
We have three important resources that enable us to integrate our approach to studying Parkinson's disease. The first of these is our biobank, which includes all of the blood samples and the DNA from our Parkinson's participants. The second resource is the neurobank, which contains over 200 cell lines derived from people with neurological diseases. The third resource is NatureBank, which is a collection of biological specimens that have been carefully collected, catalogued and prepared so that they can be used as a drug discovery resource. We have already identified 20 NatureBank extracts which contain molecules which we think protect Parkinson's disease cells from dying in culture. These may be potential neuroprotective drugs that could be used to treat Parkinson's disease.
In the past, Parkinson's disease has been seen as a single disease, in that everyone who has Parkinson's disease has the same disease. But in fact, we know that that's not true. We no longer look at it as a single homogeneous disease, but actually separate diseases. We've discovered two novel Parkinson's disease genes, which have implicated a couple of other areas of cell function which had not been linked to Parkinson's disease before. So we've kind of got some novel aspects now that we're exploring. We
identified about 15 herbal medicines that are very frequently used for brain protection that is related to neurodegenerative disorders. So our aim is to look at those 15 most frequently used medicine because they are clinically proven to be beneficial to many brain-related disease.
We rely on our information or direction as to what to do a lot depending on patient information, both clinical information but also from the cells. And so part of the Queensland Parkinson's Project is that we have a website where we're trying to reach out to as many clinicians and patients as possible to fill in questionnaires to provide information for us, and also if they want to volunteer to provide samples. That's something which is really important. In fact, it's central to the work that we do.
I have, personal contact. My father-in-law actually passed away because of Parkinson's disease. The end goal, of my research is really to see whether we can use chemical probes to understand the biology, the pathology, and for Parkinson's disease, eventually to find drug targets and to identify molecules to treat this really devastating disease.
The effort is beyond any single lab, so it's essential that we actually collaborate, and through our work here and the Queensland Parkinson's Project, we actually have a lot of opportunity to collaborate internationally.
I think we're in a very good position to identify new treatments that won't just treat the symptoms of the disease but actually the underlying biological processes. And this is what you need to effect a cure. I can see this happening in the next 10 years.